Hello, everyone. I am Praveen Bommareddy. I'm an Executive Director, Research and Translational Medicine at Replimune. On behalf of the Society of Immunotherapy of Cancer, I would like to welcome you to our Biotech virtual program series. Now, I would like to introduce our faculty today. Dr. Samir Khleif is a leader in immunotherapy with a career spanning oncology, immunology, and biotech innovation. He has held pivotal roles, including the chief of the NCI Cancer Vaccine Section and the CEO of the King Hussein Cancer Center in Jordan, now as the CEO of Georgiamune and a professor at Georgetown University. He continues to drive advancements in cancer immunotherapy. He also serves on multiple biotech boards and holds more than 300 patents in immunology and immunotherapy. Now, our other faculty member, Dr. Bruce Levine, is a pioneer in cancer gene therapy and cell-based treatments. As the Barbara and Edward Netter Professor in Cancer Gene Therapy at University of Pennsylvania, he founded Clinical Cell and Vaccine Production Facility, leading first-in-human trials and set the stage for the field. A co-inventor of Chimera, the first approved gene therapy, Dr. Levine holds 32 US patents and has co-founded biotech startups like Immunity Therapeutics and Capstan Therapeutics. You both share several key similarities in translating academic discoveries into biotech ventures. Thank you, Dr. Levine, and thank you, Dr. Khleif, for spending time with us and sharing your valuable feedback on the biotech ecosystem and your amazing journey to where we are today. I think, I would like to keep this pretty informal with back and forth discussion on, sharing your background, your experience and your transition from, academia into industry and the experience in developing these new technologies into, potentially and life-saving medicines with Chimera and other examples. So, Dr. Khleif, maybe you could go first and share a bit about your background and, in terms of the experience in developing these technologies and transitioning into biotech from academia.
Sure. So thanks, Praveen. It's great to be here. So starting from 20-some years at NCI, it was really great to be at the forefront and at the pioneering edge of the development of immunotherapy. So that gives you really a very good window towards how the whole field has been developing and evolving. So since we were there, we started working on early development of drugs and in immunotherapy. And this is how I led the first cancer vaccine program in the country, which was really a very new field, particularly targeting specific areas and specific antigens, which now we call neoantigens. At that time, we call them mutant oncogenes. So that gives you a little bit of perspective of how you think about, one, discovering something completely new that has not been tapped before. Second, breaking into breaking that field towards or into the common, let's say, field in immunotherapy, or at least in therapy in general and oncology, and then translating this into clinical trials. So that gives you the base of how you think about really knocking something completely novel, knocking something completely that's not known in the field and taking it into clinical trials. So with that in mind, under the belt, going to different institutions, including when I was the director of the Georgia Cancer Center at the University of Georgia Medical College of Georgia, started to discover and look into some new things in immunotherapy and new biology in immunology, including try to break and identify targets that could lead to the differentiation or different functionality in T cells, which means how those targets can control the plasticity of T cells. So when you try to get that, knowing how thinking about the whole process before into thinking of those targets now and translating those targets towards development of drugs that could manipulate those targets and accordingly can reset or reprogram the T cell. That was a major leap that I really learned during an earlier career, which was at NIH. So based on that, we were able to discover completely new targets. We were able to discover completely new pathways that manipulate the immune system. And with that, we spun this out into a company that the company now developed almost 11 new drugs that are purely based on those new biology that we have developed and discovered, and taking some of those already in clinical trials. So you need a lot of base to be able to think about it properly. For, again, all this from scientific point of view, from drug development point of view, and we could talk more about how you learn this as you go from an academia, but at least that's the background that led to the development, at least now, of 11 new drugs that are first in class and are in different phases of development.
I think, thank you. I really want to highlight what you said, having the strong foundation of a fundamental pathway or something interesting, novel that can lead to the development of, tens or even more of different targets or drugs. And that can be done in academic setting and then now transition to a more biotech or startup setting. Thank you for that. I think, Dr. Levine, I would like to ask you a similar question, right? Can you sort of walk us through your background, and your journey to Chimera, how you're able to achieve this in an academic setting, and any difficulties or opportunities that I think often when younger or early career scientists were in academic setting, they often under the misconception that innovating is slow, or rather not so fast in academia versus in this biotech ecosystem. Maybe you can shed some light on these things and share your journey.
Yeah, thanks, Praveen. Let me start by saying that cancer immunotherapy and cell and gene therapy in particular is a team sport. We cannot do great things without the greatness of others. And so my journey began as a postdoctoral fellow with Carl June at the Naval Medical Research Institute. And one of the projects he put me on was to develop a system for culturing T cells in the lab. It's basically an artificial dendritic cell composed of a magnetic bead to which we conjugate two antibodies, anti-CD3 and an anti-CD28. Now, this work came from discovery on elements of T cell signaling. Another element that goes into the development of what became Kymriah was a chimeric antigen receptor, and that was work of Zela Gesher and colleagues and another group in Japan. At the time that the structure of the B cell receptor and the T cell receptor were being elucidated, they asked the question, well, what happens if we graft one on the other? And then a third element would be the viral vectors, and many groups, Richard Mulligan and the developers of lentiviral vectors have contributed to that. So one can argue from three semi-disparate areas of immunology and vector biology is how we have the first FDA-approved gene therapy. Now, after we developed that method for culturing T cells, the very first group that conducted a clinical trial of a CAR T cell was actually a company, Cell Genesis, located in Foster City, California. They started their trial with a conventional method of growing cells, and they'd infuse and they'd go away. And they approached us to use our method, and we struck up a collaboration. That was our first industry collaboration. And they use our system, and now the cells engraft long-term. We come up to the University of Pennsylvania. That first trial was in HIV. We come up to Penn in 1999 to begin cancer clinical trials also continue in HIV, approached by another company, Virixis, that developed the first lentiviral vector. So we translate that to a clinical trial. And then other companies, Adaptimmune, Sangamo with zinc finger nucleases, the first gene editing. So this is a long story of serendipity and persistence and staying in academia, but having the opportunity to collaborate with biotech and now pharma to bring new technologies into the clinic. And our first CAR trials in cancer in 2010 led to the alliance with Novartis that became Kymriah. And then we begin to think, well, why are we always collaborating and striking alliances? Why don't we spin out our own company with other IP? And that became Immunity to develop CARs and solid cancers. And I'll say a little bit about the environment here at Penn at the Center for Cellular Immunotherapies, where we have 30 independent labs investigating aspects of cancer immunotherapy and T-cell biology that can be translated. We have a laboratory dedicated to the translation and scale-up. We have a manufacturing facility. We have quality control. We have correlative studies. We have regulatory support. Around 250 people altogether. That is, in essence, a mini biotech embedded in academia. So I could say we have the benefits of both, working in academia and collaborating with industry. And what that has led to now over 25, 26 years is we're spinning out companies. Capstan is one developing in vivo CAR T cells. Charisma, Interius, Verismo, Vittoria, and I serve on scientific advisory boards. So I would close with saying what has happened over the years. In the beginning, as Samir knows, we were working pretty much a backwater. No one believed in immunotherapy. And now we have an acceleration of interest, companies spinning out grants and academic programs. And we have the best and the brightest coming into the field of cancer immunotherapy now. And I think that's really beyond the scientific findings, beyond the new companies and the drug approvals, seeing the next generation come in is really, really exciting as well.
Thanks, Dr. Levine, for all the background and your thought process on an explanation on the Kymriah and your experiences. So I think what you and Dr. Khleif has mentioned is very interesting, that in both of your experience, a long journey in academia eventually led to spinning out biotechs and identifying different pathways or different technologies that can be spun out. Can you speak to the different approaches you might have taken, on a product focus versus a platform focused company? When you're developing these, companies, how did you envision if this is a product based or a platform based, and how do you know this? Right. So sometimes PD-1 checkpoint inhibitors, many companies just have one antibody, that can be based as just a product. Or if you think this is an antibody which inhibits the signals on T cells, you can extrapolate this to a platform. Maybe share your experiences on, how do you have to think differently, and when do you really leverage the product versus the platform-based approach?
Well, what you think isn't always fundable. So, and I think it also gets to the newness of this field and industry to this field. We've always approached things from a platform basis. So if we have a technology that we can bring into a trial, we might have a particular target in a particular disease. But if that works, then we translate that to the next target and the next disease. Now, 10, 15, 20 years ago, most of the people we spoke with who were investors were focused on the product. What's the product? What's the product? And we always had this vision of a platform as being more interesting and feasible, because if you have a success in one, then you could leverage it. If at first you have a failure, then you can turn to the next one. So I think our philosophy, it may still be a little different from investors, but I think we've had enough success to believe in this platform approach. That's not to discount a product focused company.
Dr. Khleif?
So Praveen, you need to first define platform, because the platform is not platform. There are at least three kinds of platform. One platform is dependent on method, like a technology. Another platform is dependent on search, which is fishing, like omics. And both of those, or either of those, could be also integrated with AI. And then the third platform is based on biology, right? So those platforms' discovery are completely different. Similar to what Bruce was mentioning is the technology platform. You have a technology, you can design or engineer, but you need to further define targets. And those targets could be defined in 3 different ways, et cetera. And this is why you said some of those are difficult to fund, because always people will say, okay, fine, where is the target, right? Particularly the second platform, which is the omic platform. We're going to do search, we're going to find something, and then that something needs to be validated, proven to be a target, and then move forward. And then there is third one, which is fully dependent on the biology, which is defining specific biological phenomenons within something and then digging into it, right? So these are 3 different platforms. The discovery and more drug development of those are completely different. And this is why when you look at companies, you need to decide which one you want to take forward depending on the technology that you have. Now, in any of those, drug development can really or will be really dependent on what target you identify and validate. Because from that now forward, it might not be purely a one type of drug. For example, cell therapy, like what Bruce was saying, is most of the approach is cell therapy. You engineer a cell, you move forward. The other type of development, if you want to develop drugs that will manipulate a target, which is different than the cellular therapy, then you need to think, is it going to be a biologic, which is a protein structure, engineered protein structure, an antibody, or is it going to be a small molecule? And others, right? So, for example, in the company that we have, which is Georgiamune, as I mentioned, we have now 11 drugs under development. Part of them are biologics, part of them are antibody, which is structured, engineered proteins, part of them antibodies, and some of them are small molecules. So it really depends then, answering your question, on how and what the best way to target that particular molecule that you identify to be able to manipulate it or change it into what you need to do, whether turn it on, turn it off, change the structure, etc. And this is why you need to be really adaptable and understand how you can think about those issues and learn. So these are extremely important.
That's great, but the question arises, how do you navigate with the investors during the time when you are constantly, thinking about product versus platform, and, what are your experiences? And also would like to hear from Dr. Levine and Dr. Khleif that how this has been evolved during the last 10 or so years, when you first started doing this to the current market, which I believe is a little picky in terms of what they are funding and what kind of new technologies they're more sought after.
Well, it changed a lot in the past 10 years. It really changed a lot. And when you have 10 years ago, 5 years ago even, platforms looked at, well, this is a good thing because you can scale up, you can reproduce. The problem with this is many companies had platforms, particularly search platforms or technology platforms, and then they had an issue identifying a validated target to take a drug towards it, right? So these things have shifted with the investors in a little bit more, okay, where's the beef? Where's the money, right? So show me that this thing can be validated and can produce a drug. So at the end of the day, what's important for an investor is, do you have a product, a drug that is validated, can work, and can be proven to be at least preclinically and hopefully clinically workable? That's the bottom line. The platform by itself is a tool, right? Is a tool towards developing this. So now, if you have a platform that's proven to work and proven to show that it could develop drugs, for example, what Bruce mentioned is clear, right? What we have two platforms, biologic platform, both of which led to the development of many drugs, and they are in clinic already. So it's the proof of concept is what is important. Now, that is also can be infectious, right? If somebody else prove it, then that might be good. But at the end, what matters to the investors now, way more than before, is that you have a drug, rather than you come and say, well, I have a platform. Well, great, but show me that you can make a drug.
Dr. Levine, do you want to add anything?
Yeah, the investor sentiment will vary depending on the economic cycle, from not interested to irrational overexuberance, right? The next shiny version of whatever has most recently been successful. I think there's another distinction that we've been wandering around that I'd like to bring up, which is the difference between a therapeutic company and a tools company or a device or equipment company. So tools company, you might call it omics, you might call it gene editing, some other technology that can be widely applicable, and maybe the company is looking to provide a service or to partner, and the therapeutic company will be stacked from discovery to translation to regulatory approval. Those are a lot more expensive. And then the question is, what do you have? How is it protected? What is the competition? And having the buy-ins from the investors really gets at that whole intellectual property and know-how package, but it also gets to the founding team. And that works both ways for the founding team to look at who the investors are, and are the investors aligned on the vision that the co-founders have for the company. And I've seen those diverge, and then that presents a lot of difficulty. When the management is on the same page as the co-founders, who are on the same page as the investors, that's the best of all possible situations to bring things forward. Of course, dependent on the success of the technology and the target.
Yeah, thanks, Dr. Levine.
Praveen, I'd like to just add one thing, just to stress on the fact that everything, again, Bruce mentioned a little bit at the beginning, but everything is dependent on the financial cycle. When there's lots of cash, there are lots of things that might not be looked at favorably in a different time than less. But what I could say is what's consistent is the presence of product that has strong validated biology and mechanism. I think that's always important.
Thank you. Speaking of the data, what kind of data, when do we think we're ready to spin an academic lab into a company, right? There's always, is preclinical data sufficient, or do we need some initial clinical data? What are the tools that are needed to really, decide or to make a decision before we say, okay, now is the time we're ready to spin this idea into a biotech, and how much data is really needed, and how this have evolved again during the last 10 years?
Well, in FDA speak, I guess it depends. So it depends on the economic cycle. It depends on the strength of the original data. And just to marry those, if we look back, say 2, 3, 4 years ago, or 5 years ago, there were a lot of companies that were going IPO based on preclinical data and huge bubbles and then crashing. So be careful what you wish for, I guess, and that there is value certainly in securing the initial funding to develop whatever you have. But once that clock starts ticking with the investors, they're going to be coming back and say, okay, when are we going to the clinic? Okay, when are you going to file? So it really takes a lot of skill and luck to be reading the data, the market, the funding environment, and the development of all of this technology. Now, at a university, we have tech transfer offices. So when an investor thinks that he or she has something worthwhile, then you approach the tech transfer office. Do you think this can be filed? What is the freedom to operate? What does the landscape look like? And then you file a provisional, and then you can file the patent. In cell and gene therapy, there are things that may not be patentable, so-called know-how, that are valuable in themselves. Let's call them standard operating procedures. That's really the secret sauce that you need to manufacture and develop a product. How those are protected are quite different. So in terms of how you know, we consult with our tech transfer office. They have contacts with investors. Since we've been doing this for a while, we have contacts with investors as well. And it's really getting a sense from the different parties. Do you think we have something? Or are we ready to think about starting a company, and what is that going to look like? Now, most new companies fail, and there's a lot of gray hair that comes with that experience and understanding what's going to fail and what has a chance at succeeding.
So I would add a couple of things, Praveen. One is, I mean, look, if you want to take something into or spinning it out into a startup, I think there are two very, very, very important issues. One, can it get funded? And second, at what value? Right? Because it's up to you how much or how far you want to take it prior to getting it funded, right? So at the beginning is, can you get it funded? And to be able to get it funded, you need two very important characteristics. One, is this a validated discovery? And second, is intellectual property protected? In any way, it doesn't have to be patent, it could be anything. So these are very, very important things. And if those are not there, it's unlikely that this could be funded. Of course, the third element, is there a market to it? Because again, you need to think about that, whether from the product itself, if there is a discovery about a product that you can buy over shelf, even if you patent the method of use, right? At the end, no one's going to fund it because it doesn't mean much because there's no commerciality to it. So scientific validation, intellectual property protection, and the potential for commercialization. If you get this, then potentially it can be funded. Then the question is, what is the value that you want to fund it for? So how long do you wait to increase that value, or go as early as possible and get low value and dilute yourself or whatever? I mean, these are the things that you have to think about.
I think another question comes to my mind really is, does this process differ based on the source of funding? NIH funding versus you have a grant from an industry. Does the source of funding dictate how soon you go into the clinic and the process of tech transfer and the difficulties and the opportunities involved in the process?
Very much so. I mean, the source of funding, and this is one of those things that any academician that is thinking of going out and spin out a company need to really understand very well, because there are categorically, there are two types of fundings. There are the non-dilutive funding and the dilutive funding. The non-dilutive funding usually doesn't come out with lots of strings attached. And those are government funding, foundation fundings, et cetera, that you can take and you can work with it as if you're working in your lab with your academic lab with specific milestones that you want to reach. But at the end of the day, it does not affect your ownership, the company structure, any of this, other than developing your product, of course, on a timeline. The dilutive funding comes out with lots of strings attached. And those include ownership, those include structure of the company, those include what's your involvement on the company. Some investors might say, well, we want you to be an advisor. Some investors say, well, we'd like you to be a CEO. So all these kind of things come into play. So that is very important to take into account, to take into consideration before the decision of when do you go out and with whom do you go out, and that affect the timeline in a very important way.
Dr. Levine?
Yeah, so the government funding, we'll see about the security of that. The foundation and philanthropic funding, many organizations now were writing in clauses where they're entitled to royalties if there is intellectual property that is licensed and it becomes a product. So while strictly not dilutive, it does come in some cases with strings attached. As far as working with industry, this also gets to what does the company become? Is it going to be independent or partner? Or is the intellectual property going to be held by the university but licensed to a larger partner? And then that gets to how much control do the investigators have over the progress of that program, right? And what we're doing in this field, we feel we have both ownership and expertise to provide. It's not like some technologies where you throw the brick over the fence and someone else can, build with that. So I can speak for myself and Samir for himself. We feel we have expertise to provide, that we want to be involved, even if there is a license to an already established company. We would like to be able to guide the development. Now, there's a tension, right, between the academy and industry. You can almost say we speak different languages, some different cultures. So there's learning to appreciate the drivers and incentives of industry with what the drivers and incentives are in academia to publish, and industry not so much. That takes compromise and understanding on both sides.
Yeah. Thank you very much for that. I think coming back to the tech transfer aspect and resources available at institutions, you two come from some of the major institutions, and there might be a lot of resources internally to help with this. So maybe you can quickly share what are the opportunities available for, any guidance for individuals who are coming from, not major institutions, some of the not well-funded institutions, and what kind of resources should they seek, and what's the pathway for the tech transfer if they don't have, internal resources that can cater to their needs?
Yeah, I think this goes back to some of my first comments, which is that doing this and spitting out companies and developing cancer immunotherapy, it's very much of a team sport. So if an investigator thinks that they're going to translate to a clinical trial, become the CEO of a company, and run their lab, it's a tall order, right? So I think the first thing is to find a mentor who has previously navigated the system at your institution, and educate yourself both by speaking with experienced peers at other institutions and educational opportunities such as this with SITC or other professional organizations, and maybe contacts in industry as well. So again, it's a necessity of multifactorial expertise. You're going to be dependent on others, and you have to understand what others have gone through to have a sense for where you want to go.
So look, this is something that we academicians do not learn in medical school or graduate school. This is something that you absolutely need to learn as you go. I assure you, you're going to make lots of mistakes. The most important is to learn from them. But the most important thing is to make sure to know who to tap into and which door to knock on and what resources to get to be able to learn the process. Because it's extremely important to learn. I mean, again, look, it depends, because some would like to have their IP and then license it and done. Some would like to have their IP and then license it to someone that would spin it out. But if you want to be engaged in it, you want to spin out your company, you want to be part of that company, you want, as an advisor or as a CSO or as a CEO or whatever, it's a completely different area. And you mentioned is some universities, they have phenomenal tech transfer offices that would provide it to you on a silver plate, but many don't. So this is why it's important for any person who would like to spin out a company to learn at least three things. One, what's an entrepreneur? And that requires advisors, that require tapping into regional offices, state offices, because they have many. Found your mentors, find some of the organization like AETM or others or SITC to try to get people to advise you. And don't be shy, because that's important. And that is not only relevant to how you think of building a company, but even how you develop a product. Developing a product is a completely different than discovering a molecule or discovering a pathway or understanding science. This is a completely different science. Again, we do not learn this. People don't learn it in medical school or graduate school. So that's another way of really understanding. How do you develop a small molecule? How do you develop a large molecule? What's an antibody requires? What's an IND? So all these kind of things are very important to learn. So that's number one. Number two is you also need, and if the tech transfer office is not as strong as it should be, you also need how to protect your intellectual property. Because many of the problems that happen is the intellectual property is not properly protected. The patent or whatever has been submitted is not complete. And then when it comes to spinning out a company or getting to investors or financing, this will have many holes. So this is why it's very important to learn of how to be able to do that. And again, you can talk to lawyers, you can talk to patent offices, etc. And then the third is also learn how to raise funds. What are the different types of funds? how you even form a pitch deck, how you put a deck together. They're very different from academic presentations. So these are all important to learn while you're going and before you step into that major field looking into it. But this is why it's important to find people and don't be shy to ask or take courses or ask people that done it before.
I think we spent some time on the tech transfer aspect, which is really important for, bringing these novel ideas into a clinic and the process involved. If there are three key things, three mistakes or three opportunities that you have missed during the initial stages of tech transfer, you have realized later when you have already done that and went into the later phase of drug development, can you share if you have any experiences along those lines, Dr. Levine? Maybe you did this several times, and how is this different the first time, the second time, and third time? As we also know, the investor feeling has been evolved during these last 10 years.
Yeah, look, I think we got a little bit better at understanding how to speak to investors, how to tell the story of commercial development rather than just scientific development, as Samir was just saying. And it's very dependent on the macroeconomic environment, how receptive people are going to be. I would like to just mention something that may be a little of a detail or technical, but being prepared in terms of organization of the data and the procedures. When you have a partnership or when you have an investor, they're looking at the pitch deck, they're speaking to the co-founder, sure, but when it gets to the development phase or the partnership phase, maybe this is more applicable to partnership. The level of organization that you have in your lab, in your program, in your clinical development program shows to them the roadmap that you've already drawn, that you know how to do this translation, that if it could possibly work, that you have a program that is going to make it work. I think that's what investors want to see, is that you have the capability to take something from the tissue culture flask to clinical trial and develop a commercial product. So mistakes, there's plenty more than three. We've talked about a lot of them. And in the end, it comes down to timing, technology, and being able to tell the right story.
If you don't make mistakes, you're not an entrepreneur. You're not doing something new, right?
That's great. I think telling the right story really resonates with me, and that's great. One thing you both have in common is that you still hold an academic appointment, or you're involved in, founding companies and being on boards and all these. How do you navigate through this? And, you can only be at too many places at once. So any advice you can give for the early career scientists who are thinking about, doing the same, having an academic appointment, and also be closely involved with the drug development or at a company?
Yeah, it takes a while to learn that, I think, with the rapid expansion of our field, cancer immunotherapy, and so many young people coming in, and then they see, okay, this PI spun out a company, that PI spun out a company. I'm a fellow or a grad student, and I have the coolest thing, and I'm going to start a company. Well, the odds are greater that that's going to fail than later on. And it is a learning experience, but it can be a very expensive learning experience. So I think, Praveen, that comes down to a great deal of self-awareness. How sure, not only how sure are you, how sure are people that you trust and have been in the field for a long time in the feasibility of what you're proposing?
So, so your question is really great, and it's an opportunity for me to actually tell everyone that if you want to do it, you can. Because lots of people tell you, no, you can't do that. This is difficult. This is impossible, etc. I don't think that if you do it the right way, you find proper mentors, you find proper people to talk to, you learn that it's impossible. It can be done. And if you can do it, you should do it. It is important because also if you can do it, then you're getting the two experiences mixed, and both of them help you a lot, which means the way you are engaged in a company and in the development of product towards commercialization will give you a lot of thought and new level of thinking of how to think about your research, and vice versa. So this is why it's extremely important. But yes, it needs to be done in the proper way with lots of backup, lots of support, and you should be 100% ready to fail and pick up yourself and do it again, or correct your mistakes as moving forward. But I'll tell you, the most important part is to surround yourself with experts, surround yourself with a great team that can give you all the aspects that you do not know, because a lot of things you don't know. And the most dangerous thing in this business is what you do not know. So this is why it's important to have the expertise around you to be able to support you. But having both and being in both, it's a very enriching experience to both.
Thank you very much. I think surrounding yourselves with the right people and knowing what you don't know, that those are some of the great advices I've heard. I want to thank Dr. Levine and Dr. Khleif for spending time with us today. At their core, both are driven by a mission to push the field forward, leveraging academic research, biotech innovation, and strategic partnerships to bring transformative treatments to patients. And to the audience, thank you all for attending the program today. And thank you both for your excellent insights. I just want to also mention that we have a couple more of these educational series coming in June. Biotech founders will discuss building companies, raising funds, and crafting a business plan. I will also lead a conversation with Dr. Robert Coffin of Replimune and Dr. Rachel Humphrey of Immunotherapy. In September, early stage investors will share how they evaluate new opportunities, support company growth, and conduct due diligence. My co-lead, Dr. Joanna Grossman, will moderate a discussion with Dr. R.D. Armau of Claiming Venture Partners and Dr. Nandita Shangari of RA Capital Management. And thanks again, and we look forward to seeing you all in the next session. Thank you.